All right, well, I think we'll get started. So, good morning, everyone. My name is McCain Vogel. I'm the associate editor here with No-Till Farmer. Just want to say thanks for joining us today for our webinar with Holganix titled Digging into Crop Residue, the Role of Soil Microbes. Presentation will feature Holganix President of Agriculture, Dave Stark. Now, Dave stepped into this role in 2015 to lead the company's efforts in the agriculture space, including commercial development, R&D, and overseeing various studies. And today he's going to be talking about soil microbes and how they can help break down crop residue, boost soil health, and improve planter performance. Now, before I toss it off to Dave, just a couple of housekeeping items here. If you've joined us before, you know some of these, but we'll go over them again. So on your screen, you should see a Q&A icon there at the bottom. You can use those to submit questions anytime throughout the presentation. And then we will wait until the end. We'll save a little bit of time to go through as many of those questions as we can get to. So feel free to just use that to type your questions, and I'll monitor that. And then just a reminder, we are also recording today's session. So in the event you want to go back and watch it later, that'll become available to you about 24 to 48 hours after the conclusion of today's presentation. And with that, I will hand it over to you, Dave.
Okay, thanks. Well, thanks, everyone. So again, I'm Dave Stark. I'm with Holganix. My background, my doctorate is in molecular biology from Washington University. I know that's kind of weird. You probably don't run into many molecular biologists. I spent 26 years with Monsanto in various roles, and after retirement, was introduced to this company that made this funky bug juice called Holganix. And they asked me to help Holganix understand agriculture, and I'm still helping them. It's amazing because I thought I was going to retire. So in my presentation, even though I run Holganix's ag business, and I'm going to show you a lot of things, pictures and data using our product, my main objective is to really help you understand what different microbes do for managing soil or crop residue and what they do in the soil. So I'm really going to focus on residue. I don't like to be real salesy, and I know you don't want me to be. So I'm just going to apologize up front that you're going to see some stuff from us. But remember, I really want to help you better understand, and so that you can make better decisions on what you want to use in your operation to control your crop residue. So now here's one thing. So again, I've been in ag technology for over 40 years now. And how many times have I said, let alone heard, that we have to be more sustainable? We have to produce more using less. And a lot of the direction that's given, that's been good direction, is, look, if you want to improve your soil and be more sustainable, you need to stop tilling, use cover crops, and over time, your soil will get better. And that's generally true. But how many of you have experienced that it's not always that easy? It can be slow, painful, expensive. Instead, what we have found is we were getting it backwards. What we have found is if you add life back to your soil, it actually will enable you to go no-till, to cover crops, to use less inputs, and improve profitability. So kind of switching the paradigm there. So again, focusing back on crop residue. There's a lot of reasons to leave residue out on the field, but you also have choices because that crop residue has value. There's animal feed, bedding, maybe biofuel production. So you've got choices to make. There's no doubt, though, leaving it out there does good things for the soil, but it's also a balancing act, because if you remove too much, what happens? You get erosion. And that's one of the worst things that happens to your ground. You lose a lot of nutrients, that valuable topsoil. You know what happens when it rains, especially after you've planted and you get crusting. You don't manage soil moisture very well. But on the flip side, in the spring, if you still have too much residue, that also can cause problems. Your soil can warm unevenly, which can mean uneven seed-soil contact. Uneven emergence, of course, it can jam the planter, slow you down. But we also know that uneven emergence and precision planting has some really good data on this. That can cost you big time in yield. So again, we want to manage the residue. Another key piece about the residue is you've paid a lot of money for fertilizer. And I don't care if it's green fertilizer, manure, synthetic, it doesn't matter. When you get down to the atomic level, it's all the same to the plant. In that residue, there's a lot of nutrients tied up. So, for example, in corn, every 40 bushels of corn produces about a ton of stover. In that ton of stover, there's about 17 pounds of nitrogen, about 4 pounds of phosphorus, 34 pounds of potassium. Potassium you're going to find in stalks and stems is really important for strength, and there's some sulfur. So your 200 bushel an acre corn crop, you got 5 tons of residue. You have a lot of nutrients out there that you've paid for. Not one tiny bit ever gets returned to the soil for a future crop unless a microbe breaks it down. So we need microbial activity to do that. So what factors influence the breakdown of your residue? Microbial activity. So the better question is, what influences microbial activity? It's a lot of the same things that impact your crop. So soil type, the microbes you want in your soil need oxygen, especially the ones that are really good at breaking down residue. pH is important, salinity, moisture, temperature, etc., are all important for microbial activity. Now, one question I get all the time is, I take care of my soil, I use compost tea or some other really diverse microbial product. Do I need to keep applying it? Yeah, the microbes in your soil are changing all the time. Some of the things that make them change, we do. Some we have no impact on. So some of the things we do, tilling, and we're going to talk about tilling, what that does to the soil. So, since you're on this webinar, presumably you're no-till, you're working toward no-till or reduced tillage, great. By the time we're done, I think we're going to reinforce why that's a good decision for you. Sometimes, though, you have to till, and I get it. But really, when avoiding it is good. Over application of fertilizer. Too much nitrogen or phosphorus signals the plant you've got what you need. Stop feeding the microbes in the soil. That changes the soil biology. It also degrades your soil because the microbes don't just die. Instead of eating what your plant feeds them, they're eating what's in the soil. So now we are degrading our soil. Also, of course, overuse of things like fumigants, fungicides. Fumigants are meant to kill everything. But, those are things we do. There are things, though, we don't do. Winter. Microbes, like most living things, are mostly water. What does water do when it freezes? It expands. That kills a lot of microbes, not evenly. Some are more freeze tolerant than others. So now we're out of whack, and balance is important, and we'll talk about that. Flooding, especially in a year like this. About 48 hours waterlogged or underwater, we start to deplete the oxygen in the soil. And most of what you need in the soil needs oxygen, just like your crops. And you can tell when that drains, if it smells like the bottom of a swamp, it's bad. You have basically killed the living biology you need in your soil. You've got some things that can survive without oxygen, and that's what that smell comes from. But that's actually not what you want in your soil. Those tend to be the diseases and pathogens. But just to make this even more complicated, yeah, you're welcome. The biome in any given root zone around the plant not only varies, up to 10%, maybe even more, between corn and soy and an oak tree, but also corn hybrid to corn hybrid, soybean variety to soybean variety, because you see, plants farm microbes. They're specific in the ones they want and the ones they don't want. Why? Because microbes mine the soil and they feed each other. So the bottom line is, The soil biome's always changing. We can't assume when we come out of winter and we plant everything is there. And here's a good example. So this is not to represent or say this is what all no-till ground looks like, because that's not true. This is an example. This is a microbiometer test. This is done by someone that shared these results with me that's in Southwest Minnesota. So what they did is microbiometer looks at fungal to bacteria ratio, which we're going to talk more about, and overall microbial biomass carbon. In other words, how many microbes you have in the ground per gram of soil. So if we look on the right, this is what we see. This is actually what we tend to see in tilled ground, but what's surprising, and this is a ground that's 15 years no-till, that's 70 micrograms of carbon, that's low. Not a lot of life in the soil, 95% of it's bacteria, so it's extremely bacteria dominant. The only thing I can think of, because this is, again, more like tilled ground, is this person uses anhydrous. And look, I know there's reasons why you use anhydrous nitrogen, but remember, anhydrous, that word means no water. Chemically, ammonia really, really, really wants to steal a hydrogen atom from water. So when it does that, and it's pulling it from something living, it kills it. Do you want anhydrous to splash on you? I hope not, because it's going to steal the water from you and cause a burn. Same thing with the microbes in the soil. Now in the center, this is a land that's been using a diverse microbial, our product for a number of years. And look at the difference. Huge microbial biomass carbon, fungal dominant. That's what we want to see. My point is, we can't assume because we're doing certain things that the soil is doing what we want. We've got to test. So I keep talking about microbes. So this is just kind of an overview, and we're going to talk about some of these things in more detail. And look, I really like to say, because it's true, this is how God designed soil to be. And if you don't know what a molecular biologist is, we're experts in DNA, molecular biology, proteins, evolution. 30 years ago, even though, look, I've known my whole life, for me, Jesus is my Lord and Savior. But 30 years ago, because of my education and such, I believe that he used evolution to create us. And I thought, Lord, who am I to tell you you're wrong? Well, he kind of laughed at me. He was patient with me. And now I'm here to tell you that actually looking into the science and the data, I will never say that again. The best explanation for the science, this is not Dave coming to Jesus. This is Dave being a good scientist and following the data. The best explanation is in Genesis chapter 1. A couple atheist Nobel Prize winning mathematicians calculated the mathematical odds of the first most simple cell arising, millions and millions of years ago, supposedly, through totally random processes. The odds were 1 out of a number followed by 40,000 zeros. 40,000 zeros. By the way, 1 trillion is 12 zeros. So I don't know what 40,000 zeros is. Any number mathematically that's the odds 1 out of a number with 50 zeros is called absurdly impossible. So when the chances of making even a small protein are 191 zeros, or the most simple cell 40,000 zeros, I'm pretty confident random is not the answer. This is all designed. So what does the design say? In your soil, remember we talked about bacteria. They're the most abundant. So in a gram of soil, how big is a gram? The size of a chocolate M&M is about the size of a gram. Not peanut, that's bigger. Chocolate M&M, that's about a gram. There can be 1 billion bacteria. They grow really fast. In good conditions, they can double every 20 minutes. There are thousands of species, many of which we haven't even identified. If we look at how much the bacteria weigh in an acre, the top 6 inches, it's anywhere from 350 pounds to a little more than 2 tons. I'm going to talk about carbon to nitrogen ratios in a minute. Actinomycetes, by the way, that picture, that's a close-up. They're really not that much bigger than bacteria. They're bacteria-like. You can see there's about 10-fold less, or 10 to 100 million, same weight per acre, fungi. Now look at that soil, all that white feathery material. Remember that picture, because that's what fungi do. They go through the soil, they're very large. That's why there's less, anywhere from a 10,000 to a million in that gram of soil. But look at the weight, 9 pounds up to almost 7 tons. That's a huge difference in soil quality, and we got to pay attention to this. And then protozoa and amoeba, far fewer in number. We don't really, the weight varies. Why does this matter? As you go down the chart, these are predators. Bacteria, among other things, are their food. So a single protozoa can eat 10,000 bacteria a day. So we don't want the numbers to be equal. We always want the prey to way outnumber the predators, but we've got to have the predators to keep the prey in balance. Why? Carbon to nitrogen ratios. And again, this is going to be important when we talk about degradation of residue and nitrogen cycling. Bacteria, for every nitrogen, about 5 carbons. Same with actinomycetes, which are very bacteria-like. What does that mean? That means these are bacteria hogs. They love nitrogen hogs. They love it. They tie it up. That's a good thing, because when you've got nitrogen in the soil, they eat it. They don't volatilize off. They don't wash off as easy in the groundwater, and they live in the root zone. It's bad because they don't let go. But now look at what eats the bacteria. The fungi, 3 times lower required for nitrogen. And those protozoa and amoeba that can eat 10,000 bacteria a day, 30 to 1. 1 nitrogen, 30 carbons. So when they're dining on this protein-rich bacteria, that's too much nitrogen for them. They got to get rid of it. So they're spitting it out. Where? Well, where do the bacteria live? In the root zone. What form do they spit it out? Ammonia and nitrate. What are the 2 forms of nitrogen your crop roots take up the most readily? You got it. Like I said, it's God's design, ammonia and nitrate. Now let's look at your crop residue and what we're asking microbes to do. So if you look on the left-hand side, and by the way, in a lot of my slides, if you look at the bottom and the center, you'll see scientific references. And that's what I like to do. I don't like just showing my data, my knowledge. I go to the peer-reviewed, published scientific journals. Almost all are from different universities around the world, and I find the data and I cite it. So that's what those citations are. So we look at manures, young alfalfa, things that produce root nodules, fixed nitrogen. They have more nitrogen in them. So their carbon to nitrogen ratios are things where bacteria can easily degrade these. And now let's look at corn stover. It's anywhere 1 nitrogen to 55 to 60 carbons. Wheat, barley, small grains, 1 to 80. So what happens in a bacteria-dominant soil, or if you're buying a product to degrade residue that only has bacteria? Where do they get the nitrogen? It can tie it up, so you can see like yellow corn. And I bet if you did a soil test, you'd find the nitrogen. You can find it inside the bacteria. It's just not being made available to the crop because the predators aren't there. So to look at it again even closer, and I know this is an eye chart, but this is important so you can see what's going on. Residue is mostly made up on a dry weight basis of these 3 most abundant polymers that are natural on planet Earth: cellulose, lignin, and hemicellulose. Cellulose and lignin are really the 2 most important that we want to talk about. Cellulose, there along the top, it's a straight chain of sugars that get linked together and linked together. And if you look at the picture here, those rods, think of rebar and concrete. Lignin, below it, is kind of an amorphous molecule that surrounds the cellulose rebar. I call it concrete. So cellulose and lignin, rebar and concrete. Lignin, by the way, is very water repellent. That's what gives residue that's strength. That's what can make a corn stalk puncture your tractor tire even 2 years after you harvest it. That's what makes a 100-foot tall tree stand up to a 90-mile-an-hour crosswind. Another thing, so if you look at the letters, they're on the right. You see little H's, that's hydrogen. You see some O's, that's oxygen, and C's, that's carbon. All these lines that are coming together, it's because scientists are lazy. It's carbon. So I see carbon, oxygen, hydrogen, and N is nitrogen, no N's. So this is carbohydrate rich. So there's no nitrogen here. So what are we going to feed it with? So now let's look at what kinds of products are being sold to control crop residue. So we make something that has over 800 species called Breakdown. It's very fungal rich. It has everything in it you need. And I'm not going to tell you what products A, B, C, D through G are, because that's not my job. And I don't like, saying bad things about people, so I'm just not going to do it. So I'm just going to give them letters. I went and I looked at other products that are being sold to degrade crop residue. There's another key point. It's in the box at the bottom with its scientific reference underneath, and I could put a lot of references here. What you want to degrade cellulose and lignin are fungi. Fungi are the only major organism that efficiently breaks down lignin and cellulose. Bacteria can do it, not as well. Some protozoa, they're specialized, can, not nearly as well. It's fungi, specifically white and brown rot fungi, not yeast. Yeast is a fungus, great for beer and wine. It's not a degrader. So what are people selling? Everything in there, except for the top product, ours, and B, are all bacteria. It's the wrong tool. It's selling you a screwdriver when you need a saw. Product B could be a good one because it says it has about 70 bacteria and fungal species. I just don't know. Do they have the white and brown rots or are they yeast? So what does this mean? This is actually a picture from the website of a product that's showing results with the residue degrader, but it only contains bacteria. So remember this picture. As you know, here the outside, the rind is pretty intact, and all these things on the inside, those are the vascular bundles. So the only thing that we're dissolving here is that white fluffy stuff, not the hard stuff. So I know another eye chart. Look here at the top at this cross section. That's looking at corn stalk crosswise. What we see, first of all, that white feathery stuff is the pith. That's what's gone here. It's the pith. Why? The pith is most of the volume of the corn stalk, but it's really low density. It has no strength. Honestly, you could spit in the middle of a corn stalk and it'll start to degrade the pith. What we need to degrade are the rind, that outside part, and these vascular bundles. What are the vascular bundles? Those are the tubes that take nutrients and water up and down the plant. The rind, the vascular bundles have 3 times the density. Why? They're made of cellulose and lignin. That's why we talked about cellulose and lignin. The pith is not. So what's so hard about breaking down residue is this outer part and the vascular bundles, because that's where the cellulose and lignin is. Now let's compare it to a product, like ours, that has lots of fungi in it, including the rot fungi. This is a photo that's taken 10 days after application. And look, you can't always tell by driving by a field if you're getting breakdown. Go out, see how it crumbles. And what you want to see is that black stuff inside, the white fuzzy stuff, that's white rot fungi, even blue. So that's what we want to see, because that is what breaks down cellulose and lignin. One way you can tell, and so you might be thinking, well, gee, what's so hard? Why doesn't everybody just sell the right biology? Fungi in particular are harder to stabilize. So our product, with over 800 species of bacteria, fungi, protozoa, amoeba, we have to refrigerate. And it's living biology. The only way we can stabilize it is to chill it down and put it to sleep. And we put it in insulated wraps on the totes so that when you get it, it's cold. You've got 4 to 8 weeks to work through it. It'll tell you when it goes bad. And this is not just our product. Any diverse product should smell good. Ours smells kind of sweet because we have a pound of molasses in every gallon. And other products you look at may as well, they may not. But if they smell like something died and is rotting, it's not diverse because it's run out of oxygen. If that happens to our product, you just call us, we replace it at our expense. You are never at risk for product being out of refrigeration too long. I don't care whose fault it is, literally. It rarely happens, but when it does, we replace it. So here's just some example. This is Iowa. This is, I have to emphasize, the conditions to break down crop residue were ideal. Remember early in the talk, I told you we need temperature and we need moisture. We had both in this field. If it were bone dry or frozen, we would not be seeing this kind of degradation. So that is something you got to think about when you are going to spend money and when you apply. Are you going to still have enough days where the conditions will give you breakdown of residue? But here, this is actually different parts of the same field. Part of it was treated with our breakdown product there on the right, part wasn't. 3 weeks later, everything was vertically tilled. Look at the difference. And there's the quote from the grower. He's basically, well, he told us he was going to sell his chisel plow. In the spring, he planted right through without any kind of spring tillage. If anything, he said, I don't think I'm even going to vertically plow in the fall because it took care of the residue too fast. Again, that's a judgment call in terms of your conditions, how much residue you've got. Vertical tillage, remember, one of the things that helps with microbial breakdown of residue is crushing and cutting. It's the same thing. Where do you get an infection? Where your skin is intact or where you get a cut? So the more we crush or cut our residue, the more infection sites, the faster we're going to get to degradation. So vertical tilling was giving more crushing and cutting, more infection sites, faster degradation. Sometimes, like in this case, it was almost too fast. Now, here's another field. Here he is. He's used the product in the spring because, again, you want to put life back in the soil after winter and in the fall to degrade his residue. Now he's applying anhydrous. And you can see the residue is looking really good in terms of its degradation. What he told us was the ground worked so well that the rows covered, the anhydrous stayed in the ground, and he could actually drive faster. Now, if you look where the tractor is pointing in the left-hand picture, the other side of the road, that's that field on the right. That's a neighbor of this guy who came over and said, "What are you doing different?" Because he was trying to put anhydrous down. And he said, "Look, the residue was a pain in the rear to go through, and my rows weren't closing." And if you're putting down anhydrous and you smell ammonia, that's your dollar going up into the air. So you don't want that. You got to get it in the ground and closed and get it to react with water so it can be stable and stop killing things. But what's going on? What's the difference between the two? And this is where I thought, I would just add a little bit to the residue discussion and tell you what gives soil structure. So this is a quote from this publication. There's a lot else that supports this, but the susceptibility of soils to compaction is a biological problem. It's a biology problem. Fewer roots, fewer fungi, and their secretions. So what's going on? Why does one ground have good structure, doesn't compact, the rows close, the ground's easy to work, and the other stuff clumps and clods? If we look closely at soil, all soil is composed of tiny particles, micro aggregates. So imagine with me 2 5-gallon buckets. In one, we're going to fill it with a really fine-grained sand. And the other, we're going to put in golf balls. We're going to take a tamper, tamp them both down tight. How tightly packed did we do the golf balls? Not much. Now let's pour water on top. It goes right into the golf balls. What it's to do on the sand, puddles on top, slowly penetrates. What we want are the macro aggregates. We want the sand to become the golf balls. How does that happen? Fungi. Fungi, which remember, especially in tilled ground, are deficient because fungi, they're a lot bigger than bacteria. They live a lot longer, but they don't like to be disturbed. So when we till ground, we kill the fungi. When we have fungi, they produce the glue that sticks all those little particles together and makes them the big size, the sand of the golf ball. Then the fungal hyphae, that white feathery stuff, and the plant roots and root hairs encompass it, like forming the outer skin on a golf ball, and they stabilize it. They protect it because bacteria are always hungry and they love the glue. So what happens when we till? We kill the fungi, we break open the golf ball or the macro aggregate. Now the bacteria can eat the glue. And then what happens? The golf ball collapses back into sand. So we want to have fungi to have soil structure. Here's a couple real life examples. So on the left, this is not far. So I live in Fort Wayne, Indiana. There's a park close to me that I run through. It's a demonstration farm. It's organic, sustainable. And around Thanksgiving last year, I had to stop and take a picture. These were the biggest dirt clods I have ever seen. Look at the face of them, tightly packed. No air. What happens when it rains? It's not going to penetrate. What happens in the spring? Well, I watched it. It was like, looked like this, except wind and it had blown in soil particles, so the hills were smoother. They had to work this ground pretty hard. Compare it to what's on the right. That's a good clod. This actually came from a customer who was showing off earthworms, but look at where the sun is hitting that. You can see the macro aggregates, that texture. You know, because you can see the moisture in that clod. And if you break it, it's just going to crumble. That's what I hear a lot of guys say, my soil is like cottage cheese. That's a good clod. That's from having fungi because it's not tilled. On the left, it's tilled and it's been tilled a lot. And the fungi are gone and they don't have the macro aggregates anymore. We can actually measure this. So we did this last summer across Iowa, 44 different farms. We applied our product at planting, again, fungal rich. And then we looked at bulk density and water infiltration over a 7-week period. Bulk density there on the top from May 15th to July 10th, about 7 weeks. Look how it's dropping. Soil is opening up. Water infiltration, 64% increase in corn, 53% in beans. That's just a difference in their root density and such. So I had one guy say, someone asked me how much rain I got. All of it. What's happening? As the fungi and the roots are growing, they're gluing the particles together, the soil particles. They're encompassing them. They're making macro aggregates for opening the soil up. So again, just a little bit of a reason why, again, I hope you understand if you're no-till, that's fantastic. You're doing a good thing for your soil. Making sure you've got fungi in there, the kind that will degrade, but also all the other kinds of fungi help with your soil structure. Remember, I talked about nutrients. So using a product, like ours, but that has the fungi that can release the nutrients, how do we measure it? Well, this is from Tennessee. So it's winter wheat, planted in the fall after corn. And then on November 2nd, he did tissue sample analysis of the nutrients in his wheat with the product and without. You can see definitely the difference in degradation of the corn stalks. Bottom line there on the left, look at the nutrient levels of NPK, magnesium, et cetera, going across left to right versus the check. Already, we're starting to see the nutrients return to the soil and going into the crop. Now, this is another way to think about when you want to control residue. It's not just in the fall after harvest, if you double crop. So these are beans after wheat in southeast Missouri. So again, we're seeing not only degradation of the wheat straw, which I've got a lot of guys tell me is such a pain when you double crop because that straw is still there in the fall and it makes harvest harder. But what he's seeing is higher emergence, and it just, well, he's also trying to show off an earthworm, which really doesn't show up. By the way, he tried different rates of the product. When it comes to microbes, more isn't always better. It's just more expensive. So, you can play around with rates, but remember, microbes are self-replicating active ingredients. So you don't always have to apply a whole lot. Another picture, again from Kentucky, same kind of deal. Double crop beans following wheat, higher yield. He also noticed because his emergence was better, he's backing off his seed count per acre because he's getting the same stand with less seed, so saving money that way too. Another way to think of what a breakdown product can do. Now, this is in Ontario, Canada. Corn last summer, planted cereal rye, and then in early June, he did his kill step. 4-foot tall cereal rye, killed it with a couple different herbicides, added our breakdown product, then planted his beans right into the cereal rye. And look, you can still see some corn residue, but not a whole lot. In that cereal rye, it's laying down, it's going away. So again, biology helps with the current crop, get it out of the ground, get it out of the ground faster, return nutrients to the soil, but also takes care of the stuff that you want to degrade. So again, when I talk about putting life back in the soil, a product like ours, yeah, we want you to use it in the spring because it'll help with emergence, nutrient use efficiency, root mass, and in the fall. For our kind of product, it's a half a gallon an acre. Apply it with enough water to get it into the soil. You can tank mix about anything but fungicide. If you're using other products that are diverse, like different compost tea type products, probably similar kind of deal. But what you want is the diversity. So again, diverse biology is going to help release the nutrients. Plants farm microbes because microbes mine the soil. So one of my strong recommendations is use a diverse microbial to start backing off your fertilizer rates. No one can tell you how much because your operation is different. But when we collect data like this and we have thousands of data points, I hope it gives you confidence that this is a tool that in your operation lets you pencil in the best trade-off between, you don't want yields to drop, so you don't want to go too low, but at the same time, almost everybody I work with can back off 10, 20, 30%. N, P, and K. I recommend doing palatable bites, 10%. That's pretty easy. And you can see after a couple applications of our product in fall and in spring, just how we're getting more nitrogen release. We're unlocking from not the just available pool, we're tapping into the total pool of P and K, and everything is looking better. Same with the crop. Healthier soil, you're just going to get healthier plants with fewer inputs. So that was a lot, and I want to give you time for questions. So just a few punchlines. To control your crop residue, you need microbes. And fungi are the key. You want to apply as close to harvest as you can. If winter is going to be a week away, so for example, where I am in Fort Wayne, by Thanksgiving, unless it's a weird fall, I probably wouldn't apply. I'd wait till spring. You want to have enough temperature days with moisture because microbes need it. Now, I lived 36 years in St. Louis. We would have plenty of warm, wet days all winter. I would have no trouble applying after Thanksgiving in that kind of climate. It's just really your call. The more days you get, the better the results. Now, some people tell me, look, I'm just going to put nitrogen on in the fall. That will help with residue degradation. If you remember the carbon-nitrogen ratios, that's true. Residue is nitrogen deficient. That's giving nitrogen to the microbes. I can also tell you Iowa State University did studies saying that nitrogen alone generally wasn't sufficient. So again, that's a call. Again, you want the biology, not just the nitrogen. Anything you can do, crush, chop, whatever, the residue helps. And tilling, if you have to, helps. But again, you're killing the fungi. If you cannot till, you're doing a good thing. So we like this picture. This came actually from a customer. Those are his kids, and they made up shirts, Holganix, so the farm is here for me. Again, diverse biology. There are other products out there, but the biology is the foundation of your soil health and everything you're trying to do. So I appreciate your attention, and I am happy to take questions now.
All right, thanks, Dave. Yeah, we've got some questions that came over, so I'll start going through those. Why don't we start with this one? Question is, how was Holganix applied at planting in the example of increase of water infiltration in Iowa?
It's a mixture. So some guys will apply in furrow and they'll mix with the starter. My recommendation is, I want you to back off fertilizer inputs. There's a half a pound of nitrogen roughly in a bushel of corn. So if you're putting on a pound or more, it's too much. Can you get to 9/10, 8/10? Yeah. But where I like to cut is pre-planter starter. And on corn, most guys are putting this in with hopefully less starter or no starter in the furrow. But for 9 years now, I've tried to figure out if you broadcast it, if you just don't have liquid on your planter, and instead you're going to do it with a herbicide at burn down or an pre-emergent or early post-emergent. Don't wait till after V3. We want it as close to when the seed goes in the ground as practical. There's no statistical difference. Again, it makes sense. Soil microbes live in the root zone. Put it next to the seed. And if you put it over all the ground, you're kind of diluting it. But remember, self-replicating active ingredients. They find the seed, they colonize, they do the job. So it's a mixture. I know it's a long answer, but I get this question a lot, and I thank you for it. It's pretty flexible.
All right, next question says, using your product to break down maize, what's the current timeline? Would this faster depolymerization allow for cover crop planting to take place during the fallow period?
Absolutely. It actually helps with emergence and stand of cover crops. So it's not going to degrade a living plant, only the dead material, but there's still a lot of microbes, even in the breakdown product, that help with germination, root development, and stand. So yeah, you definitely want to use it, especially in that race between getting your cover crop in the ground in winter. So you've got to get it out of the ground, depending where you live.
All right, got another one. This one says, I have tried your digester on winter wheat residue, but no visible difference by the following spring. Is there a way a farmer can test if the product is viable to distinguish between dead microbes in the jug versus failure due to conditions in the field?
Yeah, so it'll tell you by smell. Our product should smell, like the founder says, it's a mix between Sam Adams and glazed donuts. It's earthy sweet. When you take actually, you won't even have to take the lid off. When you walk into your barn, if you like, oh my gosh, where's the dead animal that's been rotting for a month in the heat? If it smells like that, our product has been out of refrigeration too long. And like I said, you just call us, we replace it at our expense. Another important thing, though, when you're looking at residue, I just would encourage you to go out and and feel it. Take the wheat, twist it together, see if it degrades, because remember, degradation happens from the inside out. So it's not always going to be visible on the outside. If after all that, contact me, because I'd like to know, what you saw by the time you planted. If you didn't see any improvement, I'd like to talk and learn what went on, because you should. And if not, we'll try something together to figure it out.
All right, this one's got a couple questions in one, so stick with me here. Starts out as, does Holganix offer the tests and analyses that will allow me to know my starting nutrient residue and my starting microbe load before applying your products, and then evaluate their differences during the time I'm using them?
We do, and actually we're in the middle of what we call a soil health tour. It depends where you're located, though, because we can't cover the entire country. But if you're in a place where we've got a dealer that's fairly close by, we have tools and we're going out and we're doing that because, to me, the hardest way to sell a product like this is on yield. We win with a huge percent, but yield has so many things going on, and you all know that if you don't have water at the right time, you don't have enough heat units, on and on and on, you may not get the yield. Especially, again, it's an uncalibrated yield monitor. We show you that we're making changes in your soil, and those we always win, and we don't charge you for that. So again, I would contact us because that's exactly what we want to do. We want to show you that if you're putting life back in your soil, you're making changes that are positive, that actually are going to put more money in your pocket. And so we are passionate about that and do it at our dime.
And then kind of an additional question attached to that was, does Holganix use tests for microbial changes to establish how frequently you need to add the products?
Not yet, because I'm not sure that soil science is there yet. What I can say for ground that's been using our product for a number of years is you can cut the rate. But especially in the spring, when winter just changes things, putting a little bit of the diverse biology on in the spring seems to always have a benefit. So can we ever get to a point where you just don't need us anymore? I think that'd be fantastic. But we don't know enough yet to tell you when that happens.
All right, well, we're going to conclude now. Thanks, everybody, for those questions. And thanks, Dave, again, for that presentation. Just another reminder that the entire presentation, the audio and everything will be made available on no-tillfarmer.com afterwards. So you'll get an email with that link. Usually it takes a day or two for that to come through. And then just one more time on behalf of Holganix and No-Till Farmer, thanks for joining us today. And feel free to reach out. Let us know other presentations you'd like to see us include here. And if you did have any other questions, I'm sure you can reach out to Dave directly or anyone from Holganix. I'm sure they'd be happy to help. Otherwise, you know where to find us at No-Till, and we can pass those questions along as well.
Thanks, everyone. I really appreciate your time. Have a good day.